Assalamualaikum warahmatullahi wabarakatuh. So we are from Group Aphelion. So our team members is first is Nurul Atirah binti Anwar. Next is Faizatul Najiha binti Muhammad Hashim. Next is Muhammad Wan Muhammad Adwa Haziq bin Wan Anwar Hasnan. And next is me, Muhammad Ramaikal bin Ma'ini. So our research title that we select for this assessment is antibacterial activity of black cumin, clove, and ginger essential oils against specific spoilage organism of ready-to-eat chili shrimp paste. So, so for the first, this is our poster. So for the research originality, so this research describes their methods that being used clearly, and the researcher interpret their result and their discussion. So first is research issue. So the research issue that we can get from this journal is ready to eat or RTE chili shrimp paste has a short shelf life. So according to the previous research, the paste can be refrigerated for 3 days and consumed without deterioration. So this demonstrates the limited shelf life of RTE chili shrimp paste chemical. Preservatives are used to longer the shelf life of food to prevent deterioration, and nevertheless, preservatives like butylated hydroxytoluene can be harmful to human health. So since the population is more conscious of the benefit of healthy lifestyle, the need for more green food has become a significant challenge in the food sector. So, terpenoids, phenolate, aromatic, cyclic, and alicyclic chemicals, acetonides, and sulfur and nitrogen containing compounds are just some of the components that found in essential oils. So, previous research has discovered that essential oil isolated from plants like ginger can effectively prevent uh, any microbe such as Staphylococcus aureus and also E. coli. So clove essential oils have inhibitory characteristic by inhibiting protease and amylase synthesis. So glucose uptake suppression and interference with the proton motive force. So electron flow and active transport. So the hydrophobicity of EO or essential oil is important because it allows them to enter the lipids of bacteria cell membrane, that disturbing the structure and making it more permeable, and the bacteria will disrupting the cell's growth. So for next part, the problem statement, I will pass it to Faizatul Najiha.
Thank you, Arham. Now I will talk about problem statement. The RTE shrimp chili paste is not cooked. This will cause it to be easily spoiled over time. Previous studies have reported that the paste can be eaten safely without deterioration if refrigerated for 3 days. This RTE chili shrimp paste proves that it has a short shelf life. That all problem statement. Next, I will pass to the next presenter.
Thank you, Faizatul. So the next part is about the research question. So the research question contained in this journal. The first one is, does ginger essential oil more effectively than clove and black cumin essential oils? The second one, what organic compounds exist in ginger, clove, and black cumin essential oil that help prevent the growth rate of bacterial studies? The third one, what makes the RTE chili shrimp paste easily spoil? And the last one, why are the artificial preservative don't being used in RTE chili shrimp paste? So that's all the research question. So the next part is research flow by Arham.
Okay, thank you, Adwa. Okay, so for the research flow part, So the first step is to find the raw material for this research. So the raw material that we use for this uh, research is fresh red chilies, fermented shrimp paste, calamansi, calamansi juice that we use as acid sauce, bird's eye chili, sugar, and salt. Okay, so firstly, so this the sample preparation was prepared by removing the stems of chilies before the washing process. Next, the chilies was drained for 10 minutes to remove the excess water. Then the wet shrimp paste was chopped into smaller pieces and heated in the oven at 180 degrees C for 25 minutes until it dried. The washed chilies, dry shrimp paste, calamansi juice, sugar and salt were mixed for 45 seconds under sterile condition using kitchen blender. So the RTE chili shrimp paste was prepared that been prepared was left at room temperature for 5 days and was observed daily. So the three independent batch of three of RTE chili shrimp paste were prepared and three samples per batch were analyzed. So the RTE chili shrimp paste was subjected to total plate count or TPC and yeast and mold count or YMC daily for 5 days for enumeration and isolation of spoilage bacteria in the RTE chili shrimp paste. So the first step for the first uh, research, so for TPC, 1 gram of RTE chili shrimp paste was added to 9 ml of peptone water, giving a dilution of ratio 1 to 10. A series of dilution was carried out and 1.0 ml aliquot of each dilution was plated onto the total plate count agar. In cold inoculated plates were then incubated at 35 degrees C for 48 hours. So the number of colonies was counted and expressed in CFU per gram. So for the next part, for YMC, so the 1 gram of RTH in paste was subjected to the same dilution series, which is 9 ml of the water dilution with 1:10. But it been plated onto the potato dextrose agar. So the inoculated plates were then incubated at 30 degrees C for 120 hours. The number of colonies was counted and expressed also in CFU per gram. So next part, the isolated colonies that were classified based on their morphology. A single isolate from each morphotype was selected for identification and further analysis. The isolated presumptive SSO was subjected to gram staining reaction and also biochemical test for the identification. So for the gram staining and biochemical test, I will pass it to Adwa.
Okay, thank you, Ham. So for the cream staining, the slide was firstly the slide was saturated with a crystal violet stain and left for 1 minute after the smear was created. Water was then used to rinse the crystal violet stain. The slide was then saturated for 1 minute with grams iodine solution and washed with water. The slide was tilted and flooded with 95% alcohol for decolorization, and then wash it with the water to help the decolorization and counter strain for 1 minute with safranin red. Before being examined under a microscope, the slide was washed and blotted dry. So for the biochemical test, the citrate utilization was observed using Siemens citrate agar. Glucose, lactose, sucrose, and mannitol peptone solution were used to test carbohydrate utilization. The catalyst test was performed using a slide method with a drop of the 3% hydrogen peroxide. For the oxidase test, an oxidase reagent was used, emulsifying the colony into coagulase plasma was used for the coagulase test. To perform the urease test, Christensen's agar's length was injected with colony. Finally, the step line test was used to determine the motility. And then the last one, all the result then being analyzed by statistical analysis, which is one way ANOVA. So for the evaluation of antibacterial activity on of EO on SSO of RTE chili shrimp paste, the method was Kirby Bayer diffusion method to measure the inhibitory effect of EOs. Isolated overnight bacteria were suspended into 5 ml sterile phosphate buffer saline solution and being adjusted to 0.5 mid-Fallon standards. And then the next part, 0.1 ml of diluted inoculum were uniformly sprayed on Mueller-Hinton agar plates using a sterile cotton swab. Sterile paper discs were used to impregnate 100% of 10 microliter of 3 essential oil samples, black cumin, ginger, and clove. The negative control was 10 microliter of sterile distilled water pipette on a blank disc. All pipette were inoculated at 37 degree for 24 hours. And then for the next part of hypothesis in this journal, so I can find the one is that the condition of food such as low pH, low water activity or high carbohydrate content are usually unfavorable for the growth of bacteria with some exceptions. But yeast and mold can still grow under this condition and cause deterioration of various products. So that's all the hypothesis. I will pass to the Tira for the main result and findings.
Okay, thank you, Adwa. Now I will talk about main result and findings. Table 1 is phenotypic characteristic of bacteria strain isolated from ready-to-eat chili shrimp paste. The phenotypic characteristic of isolate A to G from RTE chili shrimp paste are shown in table 1. The results show that all isolate had different reaction towards the biochemical test. Isolate A and B were gram-negative bacteria, while isolate C to J were gram-positive. Isolate C, D, G, and J tested positive for coagulase, glucose, lactose, and sucrose tests, and negative for motility tests. This could be identified as Staphylococcus spp. Um, Throughout the analysis, six genera were found in the RTE chili shrimp paste and presumed as SSO. Next, table 2 is molecular identification of bacteria, isolate of specific spoilage organism from ready-to-eat chili shrimp paste during 5-day storage using 1,6S rDNA gene sequencing technique. Table 2 show some of these bacteria were also mentioned by Stenkraus 19 and 96 to be present in the ingredients of chili shrimp paste, with the exception of Klebsiella spp, Klebsiella enterobacter, Micrococcus, Staphylococcus, and Streptococcus are expected to invade the paste from the raw ingredients as reported in Saraya. Um, for Staphylococcus spp and Enterobacter spp, there can be several factors involved, such as cross-contamination, inadequate cleaning of processing equipment, utensils, and storage in a contaminated area. Last, table 3, antibacterial activity of essential oil against specific spoilage organism of ready-to-eat chili shrimp paste. Based on table 3, gram-positive isolate were more susceptible to the effect of EOS as compared to gram-negative. This is because the cell wall of gram-positive bacteria is less complex and lacks the natural sieve effect against large molecules due to the small pores in their cell envelope. Meanwhile, the outer membrane of gram-negative bacteria cell wall restricts the diffusion of hydrophobic compounds through its lipopolysaccharide layer, thus rendering it less susceptible to EOS. And that's all result and finding. Now I will continue to research objective. First is to establish the microbial profile of RTE chili shrimp paste, and last is to determine the antibacterial activities of black cumin, clove, and ginger essential oil against the isolated specific spoilage organism SSO of RTE chili shrimp paste, and to determine their effect in the shelf life of RTE chili shrimp paste. That's all from me. Thank you.
In conclusion, RTE shrimp chili paste has been contaminated by SSO dominated by Staphylococcus spp. To control its damage, ginger EO can be used as it has been shown to reduce the SSO load of pest during the 5-day storage. Therefore, ginger EO is recommended as a natural food additive and preservative as it exhibits moderate to significant antimicrobial properties, which is a new application of food technology. So that's all from us. Thank you.